Now we are going to start with the next type of carbohydrate, which are the polysaccharides. Polysaccharides are those carbohydrates which contain many monosaccharide units in them. Typically hundreds to thousands linked to each other through glycosidic bonds. Polysaccharides have various important functions. Some of them are used to store energy. For example, starch is a storage polysaccharide in plants. Animals store carbohydrates in form of glycogen, which is another storage polysaccharide. Several types of polysaccharides have a structural role. For example, cellulose is a structural polysaccharide found in the cell wall of plants. Chitin is another structural polysaccharide which is present in the exoskeleton of insects. It is also present in the cell wall of fungi. Several other types of polysaccharides are found in the extracellular matrix of animals and plants. Broadly, polysaccharides are classified as homopolysaccharides and heteropolysaccharides. Homopolysaccharides are those which contain only one type of monomer in them. For example, starch contains only glucose monomers in it, so it is homopolysaccharide. The other type is heteropolysaccharides. These are the polysaccharides which contain more than one type of monomers in them. One such example of heteropolysaccharides is agarose, which you have used for the electrophoresis of DNA. Both of these types of polysaccharides may be unbranched or branched as presented in this figure. Let us first see the structure of cellulose. It is made up of beta-D glucose monomers linked to each through beta 1 to 4 glycosidic bond. So the bond is beta-D glucopyranosyl 1 to 4, beta-D glucopyranose. The enzyme required to break down cellulose is called as beta-glucosidase, or more commonly called as cellulase enzyme. Animals lack this enzyme. Cattle and termites have special cellulose-digesting microbes in their gut which actually mediate the breakdown of cellulose from their food. Human beings are unable to digest cellulose as they lack the beta-glucosidase enzyme. The panel on the right shows an electron micrograph. These lines are the fibers of cellulose present in the plant cell wall. In this next figure you can see the two beta-D glucose monomers of cellulose. Structure of cellulose is such that every second glucose monomer is flipped. This is shown in the figure. In addition to the glycosidic bond, the glucose monomers also have hydrogen bonds. The hydrogen bonds are shown by means of vertical blue line between the hydroxyl groups of adjacent monomers. In the lower panel of the figure, you can see two chains of cellulose. Monomers of one chain have hydrogen bonds between them. In addition, hydrogen bonds also exist between the two chains and are represented by blue lines. In this next figure, you can see that individual chains of cellulose form a sheet-like structure. They are linked to one another through hydrogen bonds. Two such sheets are shown in this figure. These sheets are stacked over each other. The individual chain of these sheets are engaged in hydrogen bonds with each other as shown by means of vertical dotted blue lines. In this way you can see that the individual chains of cellulose are stacked over each other and are linked to each other through extensive network of hydrogen bonds. Many such chains of cellulose form individual cellulose fibers, which you have seen in the electron micrograph earlier. Now let us take a look at the three-dimensional structure of cellulose. Here initially you can see two chains. These two chains are coplanar, which means they lie in the same plane. When we rotate these chains, you can see that they are in the same plane. Let us now add more chains. When we rotate the view, you can see that the chains are arranged in a sheet-like fashion, and these sheets are stacked over each other. All these chains are engaged through hydrogen bonds with one another. Hydrogen bonds are not shown in this 3D representation. The next type of polysaccharide we will look at is chitin. It is also a homopolysaccharide. It is made up of N-acetylglucosamine monomers. Now let us see the structure of this monomer. It is easy to remember its structure if you know the structure of glucose. So let us first draw the structure of glucose. Carbon number 2 of glucose contains a hydroxyl group. When this hydroxyl group is replaced by an amino group, it becomes glucosamine. Now if you take away one of the hydrogen of the amino group and substitute it with an acetyl group, the resulting molecule is N-acetylglucosamine as shown in the figure. In this next figure you can see that the individual monomers are in a beta configuration. Because their hydroxyl groups on carbon 1 project above the plane of the ring, these monomers are linked through beta 1 to 4 glycosidic bond. Note that every second monomer is inverted. The glycosidic bond found in chitin is beta-DN-acetylglucosaminoyl 1 to 4, beta-DN-acetylglucosamine. In the lower panel of this figure, you can see a beetle. The exoskeleton of insects is made up of chitin. Chitin is also found in the cell wall of fungi. Cellulose and chitin are both examples of structural polysaccharides. We will now see the structure of storage polysaccharides. Storage polysaccharides are used to store energy in the cells of living things. Plants store carbohydrates in the form of starch. There are two types of starch. These are called as amylose and amylopectin. Amylose is an unbranched polysaccharide. It is made up of alpha-D-glucose monomers which are linked to each other through an alpha-1 to 4 linkage. Amylopectin is also made up of alpha-D-glucose monomers which are linked through alpha-1, 4 glycosidic bond. Amylopectin, however, is branched. In addition to alpha-1, 4 bonds, amylopectin also contain alpha-1 to 6 glycosidic bond. These alpha-1 to 6 linkages give rise to branch points. As you can see in the figure that carbon number 1 of this glucose monomer of one chain is linked to the carbon number 6 of the other chain through glycosidic bond. So both amylose and amylopectin have same monosaccharides in them. Amylose is unbranched, however amylopectin contains branches in it. In the figure you can see the starch granules present in the plant cells. Plants synthesize glucose through photosynthesis. Excess of this glucose is stored in the form of starch. This next figure shows the arrangement of glucose monomers in starch. As you can see here, the bond angles between the glucose monomers are such that it forms helical structure. Complete digestion of starch required several enzymes. The enzymes which break down starch are generally called as amylases. Glucosidase is an enzyme which breaks down starch to release glucose monomers. There are also special enzymes which are needed for breaking the alpha-1 to 6 linkage. These are called as debranching enzymes as they remove the branches by breaking down alpha-1 to 6 glycosidic bonds. Glycogen is a storage polysaccharide in animals. It has structure similar to starch. Sometimes it is also called as animal starch. Glycogen is also made up of alpha-1, 4 glycosidic linkages between alpha-glucose monomers. Glycogen has an extensive network of branching. The branch points have alpha-1 to 6 linkage. However, glycogen is much more branched as compared to amylopectin. Figure below shows the structure of glycogen. The yellow circles represent alpha-1 to 4 glycosidic bond between glucose monomers. Red circles represent the branch points. The figure on the right shows micrograph on animal cells containing glycogen granules in them. The figure on the right shows micrograph on animal cells containing glycogen granules in them. When the animal cells need glucose, they break down of glycogen. The enzyme which breaks down glycogen is called as glycogen phosphorylase. The enzyme breaks down glycogen to release glucose 1-phosphate, which is then converted into glucose 6-phosphate and can be utilized through glycolysis. This table in the next slide gives an overview of various important types of polysaccharides. Starch contains alpha-1, 4 linkage between glucose monomers in case of amylose. Amylopectin in addition also contains alpha-1 to 6 glycosidic bonds. The branches are found after 24 to 30 glucose monomers. Similar to starch, glycogen is also a homopolysaccharide. It has similar nature of glycosidic bonds as found in amylopectin. It contains branches after every 8 to 12 glucose residues. Cellulose is an unbranched homopolysaccharide. It contains beta-1, 4-glycosidic bond. Dextrans are structural polysaccharides found in bacteria. They contain alpha-1 to 6 glycosidic bonds in addition to alpha-1 to 3 branches. Peptidoglycan is a structural heteropolysaccharide found in bacterial cell wall. Agarose is a heteropolysaccharide found in algae. Hyaluronate is a structural heteropolysaccharide found in the extracellular matrix of animals. We will look in the structure of peptidoglycan and hyaluronate in the coming sessions. Cellulose and starch contain glucose monomers, but their structures are very different from each other. Cellulose contains beta-glucose whereas starch has alpha-glucose monomers. Also in cellulose every second beta-glucose is flipped. So because of steric clashes, pattern of hydrogen bonds and the configuration, the bond angles are different. These factors are important for governing the structure of these polysaccharides.